As living organisms reproduce and grow, they're constantly replicating DNA with a high degree of precision. However, inevitably, about once in every billion nucleotides that are replicated, an error occurs in the replication process, changing the sequence of nucleotides. Chemicals called mutagens can increase the chance of such mistakes. There are two major categories of mutagens: radiation mutagens and chemical mutagens. Different types of radiation have different effects on the DNA. Ionizing radiation, such as X-rays and gamma rays, can cause some of the molecules within cells to lose electrons, becoming highly reactive ions and free radicals. Some of these reactive ions and free radicals can combine with bases on DNA, resulting in errors in DNA replication, and therefore mutations. Even more seriously, these groups can react with the sugar phosphate backbone of DNA, causing breaks in chromosomes. Non-ionizing radiation in the form of UV light is also mutagenic because it can cause adjacent thymine bases to covalently bind to one another, producing thymine dimers. Such dimers can cause serious harm or death to a cell if they're not repaired, since these dimers prevent the cell from properly transcribing or replicating such DNA. Chemical mutagens have many different methods of causing mutations. Let's look at how three different types of chemical mutagens work. Nucleoside analogs are compounds that are structurally similar to normal nitrogenous bases, but with different base pairing properties. These compounds can become incorporated into growing DNA during replication, replacing their related base. Once incorporated, the nucleoside analog can inhibit further replication or cause mismatching in a future round of replication. For example, 5-bromouracil is a nucleoside analog of thymine, but it often pairs with guanine rather than adenine. Incorporation of 5-bromouracil can therefore lead to a base substitution mutation of a guanine for an adenine. Some chemical mutagens directly alter the structures of the nitrogenous bases of DNA. For example, nitrous acid can chemically alter adenine bases so that they pair with cytosine rather than thymine. During replication, this change causes a base substitution mutation in the daughter DNA. Some chemical mutagens cause small insertions or deletions of nucleotide base pairs, which can lead to frameshift mutations. Examples of such frameshift mutagens include acridine, a dye commonly used as a mutagen in genetic research, benzopyrene, which is found in smoke and soot, and ethidium bromide, which is used in laboratories to stain DNA. These chemicals are the right size and have the right chemical properties to slip between the stacked base pairs of the DNA double helix, producing a bulge in the molecule. During DNA replication, this bulge can lead to the insertion or deletion of one or more base pairs in the newly synthesized DNA.